OK, parfait.
I think if you sit somebody down and say, you have to be creative, where do you start with that? I think that's so overwhelming. You can't just force somebody to be creative. One strategy was to have this series in our main lab meetings. It was called the Big Idea series. One person speaks every week, and at the end of the lab meeting, they present 2 slides on a big idea. And sometimes the ideas are absolutely bonkers, and we encourage that and we support it. So that's the space to just go wild and be creative.
Welcome to the Night Science Podcast,
where we explore the untold story of the scientific creative process.
We are your hosts.
I'm Itai Yanai.
And I am Martin Lurcher. Today with us we have Sam Morris. Sam is an associate professor of genetics and development at Washington University in St. Louis. She did her PhD at the University of Cambridge, investigating fate decisions, and then moved to Harvard Medical School, where she worked on cell type engineering. In 2015, Sam started her own lab, where she researches how development is reprogrammed, including how regulatory genes drive differentiation and developmental programs. And she does this through computational and through experimental studies.
And basically, Sam's trying to dissect out the changes that lead to one cell assuming a particular identity. So in studying how embryos develop, she's actually elucidating the map of the possible developmental decisions. And she's obsessed with navigating this space of cell identity. It's important because it allows her to think about medically relevant issues and how we might repair or replace disease organs. She seeks to engineer cells to not only transplant back into patients to treat them, but also to use the engineered cells as a tool to study disease and development in the lab. And Sam is also a contributor to the big consortium of the Human Cell Atlas, which is an exciting initiative that's creating a high-resolution map of every cell in the human body. Welcome, Sam.
Thank you. Great to be here today.
No, it's great to have you.
Yeah. Sam, I'm wondering if you can reflect back on your early training and how it is that you got a glimpse of what it means to become a creative researcher.
Yeah, so my early training, I was the first of my family to go into academia, really to go to university. So it was not a clear path to me. It wasn't really obvious how I would achieve this. So I did my undergrad in London at Imperial College. That was on biochemistry. And I had this amazing dev bio lecturer called Jane Safel. And I think she could see how interested I was in the subject because she said, hey, why don't you come join my lab for a summer? And that was my first lab experience. And I absolutely fell in love with it. And the research wasn't very hypothesis driven. I just loved getting my hands on the experiments, and that really planted a seed. And she said, hey, you should think about doing a PhD. And it went from there. And I ended up in my PhD, and that was a little bit of a failure to launch in the PhD process. It wasn't the right fit for me. I didn't get the support that I needed scientifically or creatively. And at the end of that 3 years, you have to do your PhD in 3 years in the UK, which is insanely fast.
That is insanely fast.
Yeah, I remember.
For the first 2 years, I was completely lost. It's like, here's your bench, go. You've got 3 years. We'll get fined if you don't finish in 3 years, which I don't think was true, but the pressure was there. And I just did not respond well to that. And by the end of the first 2 years, I kind of started finding my feet and fell in love with my project, but it just didn't take off. And then I saw an advert for a postdoc position in Magda Zniczka-Goetz's lab in Cambridge. And this was on live imaging pre-implantation mouse development. And I thought, wow, that sounds incredible. Magda had an incredible reputation at the time. And I said to my PI, I want to go do this as my postdoc. Will you write a letter for me? And he wrote in my letter that I wasn't very creative. I'd never invented
Oh,
anything.
Oh,
So this is a great subject in terms of creativity.
seriously?
But he said, I'd never invented anything in the way that she had, in the way that Magda had invented things, because, she's very creative in terms of live imaging and the approaches she took. And he
Uh-huh.
came out to me and he said, I've done a bad thing. And he confessed he'd written this terrible letter for me. So we had a bit of drama, try to fix that. And I finally met with Magda and I fell in love with that project. And it's this whole process of falling in love with projects, right? You have to love it. And she told me she saw straight through the letter and she'd seen something in me that she thought could work. And so she gave me a huge opportunity. And I think I was really trying to prove myself at that point. I didn't do a great PhD. I didn't publish particularly well from that. I didn't have much confidence, but here was somebody who's willing to give me a chance. And getting thrown into Magda's lab at that time was amazing. It's such a creative environment. So we were working with pre-implantation mouse embryos and trying to do this non-invasive live imaging and lineage tracing. So this is really where I got my start doing lineage tracing. And these experiments took such a turn. So we'd ask, well, what if we take these two cells from completely different embryos and smush them together? Or what if we count the number of cells in this embryo and image it and then transplant it back into a pseudo-pregnant female? How will it develop? And so it's this constant kind of invention. And
yeah, that sounds crazily creative.
it was. And Magda, she'd come up with some of these ideas and she's like, oh, well, why don't you trace these cells and then take them and do single cell RNA-seq on them? So this was back in 2007. So this was with Fushu Tang's protocol.
Yeah, Tang it all.
Yep.
Yep.
So Azim Zarani's lab was just across the hallway. So it's a perfect mix of people who are crazy enough to try, staying up at night, tracking these cells and dissecting them from an embryo and, really trying to understand how the origins of those cells contributed to their future identity, and then trying to recover them and do single cell RNA-seq with Fushu and, mouth pipetting these single cells into Eppendorfs. And we did 3 cells. It was something like 15,000 pounds. It never got published. And it was just so many fun experiments like that. And I'd go to Magda and I'd say, Magda, this is not going to work. It's physically impossible for me to do this. This disobeys the laws of physics. And she'd be like, nonsense, nonsense. And she would get in the lab and get on the microscope, get hold of the mouth pipette, and she'd show me, see, you can do it. Like, try again, try again, try again. And it was this process of just trying things over and over. And then some kind of magic happened where you would learn physically how to put these cells together and how to manipulate them, or how to, squish cells in a mouth pipette and, really put different pressures on them and learn how the embryos respond to your manipulation. And it was this process of playing, failing, and having confidence that something would come out of it that led to a lot of creativity in that lab. So I feel like that's where I kind of learned my creativity in science within Magda.
Yeah, and that was quite an impressive transition, right? From your PhD, where at least according to your supervisor, you were totally non-creative, to a project where it sounds like, all you did was creative stuff. Where do you see yourself now in that spectrum? Like, how much of your scientific work is creative and how much is just working through the protocols?
Yeah, obviously the theme of lineage tracing has continued through my career. So I started in Magda's lab with the live imaging. I'd actually started doing some live imaging and Xenopus development as a grad student. But these were experiments that my PI didn't know about. So I think this is why he hadn't seen my creativity and appreciated it. So I should have been more confident in telling them about these experiments. So it went from, tracking cells in frogs, lineage tracing in mice, and now we're developing new genomics-based lineage tracing technologies across a range of systems. We think about the biological problem first. So I'd say we're grounded in a good biological question. That is, how can we measure and manipulate cell identity? Around that, we're interested in how transcription factors control cell identity. So, lots of nice hypotheses there that the NIH will hopefully like to fund. Really important to have that. So that grounds us. But then around those questions, we're constantly asking, how do we develop new tools to generate new data sets? What are we missing from these data sets? How do we go a step further? And then how do we develop these new technologies? And then how do we enable other groups to be able to apply them to their problems? So ultimately, we want them to have broad application. So I'd say it's a good mixture of hypothesis-driven research and playing with the technology. And I know the lab is probably 75% people who are driven by the technology development. And I know at times the other 25% of the lab have felt like, well, we're kind of overlooked in this lab because we think about hypothesis, we think about the basic biology, but Sam's always interested in a shiny new toy. So I've been really careful to address that, and every so often we'll bring it back around to the hypotheses and read a lot of papers. We share a lot of papers, and I encourage not to just focus on the new technologies, but actually focus on the biological questions as well. So to try and keep it balanced.
Yeah, so you described it as a balance between technology-driven projects and hypothesis-driven projects. But at least in my experience, and I think the same may be true for a lot of other scientists, you start with a hypothesis, but typically you find out, well, that hypothesis doesn't work out and you have to modify it or come up with something totally different. So when you say hypothesis-driven, how does that play out?
It plays out quite frequently. I'll be giving a talk and I'll say, well, we came into this project with this hypothesis and we were completely wrong, or back in 2014, we came to this conclusion based on bulk expression data, and it turns out we were completely wrong. I think it's important to reflect on how you've come to conclusions previously based on certain technologies and being able to adapt to new information, basically. I think that's incredibly important, and our hypotheses are evolving all the time. I think one example would be we applied our lineage tracing approach to one of our favorite reprogramming systems, so converting mouse embryonic fibroblasts to these induced endoderm progenitors. And we found these two trajectories of reprogramming. So one trajectory went to a successfully reprogrammed state, and there was another trajectory to something we called the dead end state, because these cells started to appear as though they were falling back toward a fibroblast identity. So we said, oh, we'll call them the dead end. That's really snappy. And we're coming to realize now that they are themselves a partially reprogrammed state. So the term dead end is probably not too accurate. So now through the literature, through the publication and pre-printing process, and also through my public communication of our results, I'm going back and I'm addressing that and saying, okay, honestly, before we called these the dead end, we realize now that's probably not so accurate. We think we can actually take these cells and then push them into a successfully reprogrammed state from this dead end state. The project's constantly evolving. I think you have to take in new information all the time and also admit when you potentially misrepresented something, because I think the language and the semantics are really important for communicating these ideas, and we don't always necessarily get that right, I think.
And Sam, when you're following along with the project and steering it as new results are coming in, it seems just from listening that confidence plays a big role, that you've sort of developed the confidence to trust your instincts. Do you notice that over time you're becoming bolder?
Definitely. I think a fantastic question, and it's something I mentor my students, or I try to mentor them in. Having the confidence to attack a project, also having the confidence to see it fail. I had that experience in my postdoc that was almost magical. You just try something so many times, eventually it will start working as you gain experience. Sometimes you need to know when to cut your losses, and that's always a difficult conversation. But I always say to my students that they've got some really challenging projects, and sometimes they lose their confidence with it. They worry if they'll ever graduate, or can they have a safety net project? And they always have a safety net project in my mind, even if they don't necessarily realize it. but I'll say to them, if this were easy, it wouldn't be worth doing. It's difficult. It means that other people will struggle with this. So if you get it to work, you've achieved something that's going to be fantastic. And also the students are quite worried about being scooped. And I also say to them, if this is so difficult, other people aren't going to be able to do it so easily. So it could give you a little bit of protection as well. So yeah, every time there's a failure, I will say, if it were easy, it wouldn't be worth doing.
My friend Katerinaki Rakusi, when I was a postdoc, she was a postdoc too, we used to say that in science, only bad things happen fast. Which perhaps is pertaining not only to science.
So just now you were talking about mentoring students to have confidence. Do you also consciously mentor them to be creative?
I do, and I do that as part of a team effort, because I think if you sit somebody down and say, you have to be creative, I mean, where do you start with that? I think that's so overwhelming. You can't just force somebody to be creative. So the way we've done it in the past, we take several different strategies. One strategy was to have this series in our main lab meetings. It was called the Big Idea series. One person speaks every week, and at the end of the lab meeting, they present 2 slides on a big idea. And a lot of that will be technology driven. So saying, well, hey, what if we could, capture lineage information with single cell ATAC-seq? And, that project's, really getting there. And that was a part of the Big Ideas series. And sometimes the ideas are absolutely bonkers, and we encourage that and we support it. So that's the space to just go wild and be creative.
I love that. The Big Idea. And it has to do with their own projects or even anything unrelated too?
Anything unrelated. If they have an idea on somebody else's project, they're very welcome to come in and do that. And the way I structure this is everybody in the lab has their ownership of their project. So I don't put people on competing projects. I try and give them space so they don't feel like their toes are being trodden on. But then I create overlap between the projects and overlap expertise so that somebody can come into somebody else's project at a time when they need help and say, well, hey, I have this skill set, I'd like to apply it to your project. Or, they can go to someone and say, I need your knowledge in this computational area to help with my analysis. Where can we go with this? And I found that's great for creativity, and it also helps people contribute to other people's projects and come up with ideas. Mm-hmm. So often I'll come into lab and there's, the classic whiteboard with the markers, and there'll just be, hieroglyphics on there. I'll be like, what have you been coming up with here? And, a lot of the time it doesn't take a solid direction, but a lot of the time it really helps.
I love that, because I feel like when you want people to work together, you have to first stabilize the situation so that no one feels threatened, so no one feels as though they're directly competing with someone else. And you also want them to be close enough that they each have a little skin in the game, that they are invested in it. Do you actually do it organically in the lab, where you see how personalities get together, or do you engineer it by putting 2 people that you think would get along, even though they haven't decided yet that they get along?
It's a little bit of both, and always depends.
It's like matchmaking, right?
Yeah, the speed dating almost. People ask me what my mentorship style is, and I always say it depends on the trainee. It differs all the time, and sometimes I've teamed people up and I thought, oh, your expertise is absolutely perfect, but then the personalities don't fit quite so well. So we go through that matchmaking process, and it's great with the students because they come in, they do, a few weeks, a couple of months rotation, and you can see how they perform. So I don't necessarily give them a set project at that time. I'll just give them an area to work within and people to work with, and then I can get a feel for, how that's working out. And before I offer, extend an invitation to join the lab, I'll sit down with each lab member and get their feel for that person. How would they fit in with the lab? How would they work with them? And that feedback helps me decide where to place them, given their expertise, given what they're interested in. And that's an important thing to note. Some of our students come in with amazing first author papers and they've got years of research experience. Some students come in with almost zero research experience. But just their personalities and their willingness to work as part of the team, for me, is more important than that experience, I think.
So it sounds like you're very structured and very conscious in the way you mentor the people working with you. But is that maybe because you had similar experiences in being mentored in the past, or are you actually being mentored right now by other people in your department?
Yes, I think a lot of my mentorship style stems from my PhD and my postdoc. I had very good examples of how I didn't respond well to a certain mentorship style. That was an absence of mentorship, and a lab where there was bullying, just a terrible, terrible environment and issues that weren't addressed. And then moving to an environment where I was closely mentored and flourished as a result of that. So from my bad experience, I make it very clear to the lab that, I have zero tolerance for any form of bullying. If there's any tension between people, we need to sit down and talk about it and address it. From my good experiences, I try and take that good mentoring style and apply it. In terms of my own mentoring, when I first started my lab, my chair, one of my chairs, I have two, she had the structure for formal mentoring. So it was mentoring by committee, and it's worked really well for a lot of PIs in the department. And it's great. You're a new PI, you're really desperately trying to get a hold of the ropes, how to run a lab. And so they sit you down with 5 or 6 hand-picked senior faculty members from across the university. And they go through your research plans, what you've achieved. It did not work for me. I found it such an anxiety-inducing experience because every single person had different advice. It was all conflicting and it panicked me. And I said to my chair afterwards, I didn't like that. It's the most anxious I've been running my lab. It's making me question everything in a really bad way. And at the time, she was concerned. She said, you can't not have mentors. And I thought to myself, wait, I've got 2 fabulous mentors on campus. So one is Rob Mitra. So he's in the Department of Genetics. He developed the calling card technology. We ended up collaborating together. And that early phase of setting up my lab, he was such a huge support to me, really encouraging, and pushed me and told me not to wait around for people to help me. He said, you've got all the skills you need, go for it. So Rob's been fantastic. The other mentor was Ben Humphreys. And so he's chief of the renal division. Oh, I hope I'm getting that right. So he came onto campus, was really interested in setting up single cell technologies. I taught his lab how to do DropSeq, and this collaboration flourished from there. So he does a lot of single cell analysis in human kidney organoids. So fantastic scientist, but he would always give me practical advice. And Ben and Rob are still around and I can go to them for advice now. Back then, they were on hand to, I could just pick up the phone and chat to them. And they'd be straight talking with me, which I loved. And that worked for me. So I said to my chair, hey, I've got these 2 people and this mentor-mentee relationship has just naturally evolved. And I think it's much healthier for me. And I actually think they give me really good advice. It wasn't always the advice I wanted to hear. And it wasn't always advice that I agreed with. So it wasn't just like they were telling me what I wanted to hear. It could be really challenging sometimes. But I've expressed several times that I think it's important to find mentors like that throughout your career. And as I look back, I've always had some kind of, spontaneous mentor, a peer, which I've been really lucky to have that. And I encourage people to find that. I think more recently, I've been kind of finding my mentors more externally.
Yeah. And, Sam, listening to you really strikes me that really you're describing first an experience where you had a committee of 5 or 6 senior faculty members telling you, reviewing your work in a very formal, in a distance and hierarchical way. And then what you found much, much better, less stressful, is kind of working one-on-one with a mentor and then also one-on-one with another collaborator, really. And so it makes me think that it's sort of like the same mentoring style that you might have with your lab, where it's, less hierarchical but more individual one-on-ones. That is a better mentoring style. But my question to you is, though, do you see some kind of need for, in Hebrew we call it in Hebrew we use the English word distance, we say it like that, distance, where distance means that you maintain a kind of hierarchy between you and another person because of seniority, or think of, like, in the army, a captain and a, what's below a captain, private. And there's advantages to having distance. Do you find that being a friend model more suitable for
I made that mistake early on, trying to be a friend and oversharing with my lab members. So they were rotation students at the time. And in terms of oversharing, it wasn't any personal life oversharing. It was just oversharing all the stresses of getting a new lab up and running. So when I was not pleased with reps or equipment or all the problems, just the workload in terms of administration of running a lab, I'd share too much of that with those students. And they didn't need to hear that, but then it wasn't good for my leadership, because I need them to focus on the science and not worry about what goes on behind the scenes. I'm here to fix problems and enable them. And to share those problems with those, I think, really eroded the leadership. So I ended up not taking any students into my lab that first year. And my mentor, Rob Mitra, was horrified. He said, "Sam, you've got to take somebody. It's terrible. You'll get a reputation for not taking students." And I said, no, that was a false start. I'm going to wait. And, I've learned something about my own leadership style. That's a steep learning curve, to learn how to lead, constantly adjusting it as the lab grows and as the makeup of the lab and the personalities change as well. That needs to continuously evolve.
So when you talk to the people in your lab now and you need to solve problems with them, do you tend to do that with groups of people, or do you tend to do that with individual people? I mean, just like you prefer to be mentored by individual people rather than a committee.
Yeah, so we tend to do it individually on a kind of daily phase of the project. So I have meetings with each group member once a week. They have their 30-minute slot. Some of them have a slightly longer slot, depending on where they're at in their projects. And that's a one-on-one. Sometimes we talk intensively about science. Sometimes we talk about food, vacations. It's just a chance to connect with people, which I love. And we talk about the specifics of projects. We also talk about lab dynamics a lot. I've got a few go-to people who will tell me when the lab dynamics are slightly off, or when they think somebody's struggling and they need attention. They're my canaries, and they're fantastic. And in terms of group mentoring, I use that approach when we are bringing a project to its conclusion. So when we're coming close to submitting a manuscript or putting out a preprint, then we'll bring the larger team together. And I think it takes the stress off of that one person feeling like they're being pecked at and being told to, you've got to do this faster, or this is wrong, or we need to fix this. It emphasizes the team and how each person can contribute to the project. So it really depends on the phase of the project. And each paper that comes out of the lab, you see at least 50% of the lab members on these papers, because it's interdisciplinary and we need so many different sets of expertise to make these projects happen. Even if it's just, some kind of fish assay that we've done, or, a small part of the analysis, people come in, do their bit, get authorship on the paper, but more importantly, they become part of that team and become invested in it.
So now, Sam, I want to ask you about a given paper that's being developed in your lab. How is it actually created? Is it a draft that's getting emailed back and forth? Is it a Google Doc that people are working on simultaneously, but maybe in different rooms? Or are you sort of in the same room, bouncing around ideas and hoping to discover something new as you're writing the paper?
Again, it depends on the person. So we have 2 manuscripts at the moment, and they've synchronized perfectly. I saw the lab do this to me on purpose. Okay, she's going to be really busy with grants right now. Like, here are a couple of manuscript drafts. I am honestly the happiest when I have manuscripts to work on. I love that process. I think it's really an important part of my creative process. But it depends on the trainee. One of my trainees, Wenjin, she's a student working on cell type classification, essentially. And we just bounced back a Word document on Slack, and I'll do a bit of editing. I'll shoot it back to her. She'll do a bit of editing. And then the other manuscript right now is for Cell Oracle. So this is a project led by my postdoc, Kenji Kawamoto. And this is our network biology approach to simulate transcription factor perturbation and attempt to predict the phenotypes of mutants and of transcription factor perturbations. And that project, we pre-printed it last February, and it's gone through one round of review, and we've been revising it for about a year now. And it has been such a fun experience. And Kenji and I have such a different working style to the structure that Wenjin and I have. So Kenji and I work a lot on Google Docs, and he prefers to communicate verbally. And so I'll edit the document, and he'll say to me, "Okay, Sam, we need to talk on Wednesday. There are a few issues here." And he'll pull me up. He's like, "I don't like the wording of that sentence." And he thinks deeply about the meaning of each sentence that is constructed. And sometimes I do that PI thing and go a little bit overboard with the claim. And Kenji will reel me back in, and we'll come to agreement on the language. That's been a really fun project, and it's benefited from the review process, because the review has really challenged us to think more deeply about how we were presenting some of those results, how we were quantifying them. And it's just helped that project evolve in a really fun way. And, Kenji came in, I just thought he was a fabulous scientist. I don't remember giving him a specific project to begin with. I think I did, but he just kind of went off in his own direction, and it was incredible. So I was like, okay, I will support you wherever you want to go with this. And then eventually came back to me, and we started working on that creative process together. And it's been a lot of fun.
So you were saying that working on a manuscript is your favorite part of the creative process. But can you explain that a bit more, like how this creativity unfolds?
Yeah, I sit down to a manuscript. I love it now because I tend to not have to start that whole process. I get a body of text to work with, because it's always so difficult to put that first draft together, right? And then I start thinking about the project deeply. I start putting it into the context of the current field, the current literature, and trying to identify where the holes are in the story, what needs more attention. But then the really creative part for me is the discussion. And I love writing discussions, because then you write that section on, well, our current approach is limited in these respects, or this current study is limited because we don't have this technology, and it's possible for us to do this. And then that starts planting seeds for new projects in the lab, and also starts setting the next direction. And then that feeds into the grant writing process, which I also find part of my creative process. I hate writing, or I think I've learned to love writing, but I used to absolutely hate it because I procrastinate wildly. I definitely got faster at writing, but I didn't write a grant for about a year. And then I got back to the process. I was dreading it. I was like, oh, I don't want to write grants again. And then I sat down and I got so excited about all these directions we could go in. And so I realized that the writing process is central to creation in this lab.
That's interesting. It's as though a lab member of yours will send you the answers, and you will think really hard to figure out what are the questions, right? Like this is the data, this is what has been observed, and you're trying to put it in the context of what does this mean in light of what we know.
Yeah, and then sometimes we'll try and flip it the right way around and go in with a hypothesis. And this is where the thesis proposals are just so valuable. And getting my students to write their thesis proposals, I lose count of the times I yell like, "Where's your thesis proposal?" And so important is sit down, generate your hypothesis, get in front of that committee, but let it guide your project. So those projects are very hypothesis driven. I think it's really important because you can also fall into that dangerous area where you have the data in front of you and you manipulate it into a narrative that's not reflective of what that data is actually telling you, or you can misinterpret the data in a really dangerous way and let it bias your experiments downstream. And so being really careful about that. So often trainees, I'll have this, oh, we can frame the narrative like this and do these next experiments. And they'll be like, hold up, think about this a little bit more objectively. So trying to strike that balance is really important because you don't want to influence a trainee's downstream analysis in a negative way because you are pushing a particular narrative that would be good for a grant or good for a paper. And I've definitely experienced that before when I've had a PI say to me, wouldn't it be great if the results showed this? And it's like, that's dangerous. And so trying not to push that, but also trying to create, these stories that people want to read and they're excited about them as well. So this whole public relations strategy, being a PI, is you wear so many hats, right? But I think you have to be careful with that.
Yeah, but when you have the data in front of you and you explore it, which I think, or Itai and I both think, is a very important part of the scientific process, then of course you have to be careful in how you interpret that data that is giving you the idea. But if you then devise some independent tests of the ideas that you just got from the data, then that would actually be okay, right? Because it sounded like you were suggesting that would be dangerous. But what would be the danger?
I think if it influences the way you're analyzing data so that it biases you toward a particular outcome, I think that can be dangerous. If you begin to overlook anything in the data, if you suffer from confirmation bias, for example, I think it can lead you to be more subjective about your analyses and say, well, I'm just going to do these analyses. Oh, I got the answer I was looking for. I think in single cell biology, the classic example is putting together that UMAP, right? And oh, well, it has this, or do some pseudotime analysis and then derive too much meaning from a particular visualization of your data. And then the way you narrate that and then the experiments you design downstream could be quite biased toward a particular outcome. So I think that's where it can get risky. I completely agree that the orthogonal analyses are essential. So I absolutely love exploring the data myself. I always encourage my trainees, and I do this myself, actually take a look at the raw data. What is it telling you? And this was so valuable for our clonal analyses because, all of a sudden you could see the signal, the noise, and we figured out a strategy for dealing with that just from looking at the raw data. You get that gut feeling about something. But I think validating things orthogonally is always the way to go, just to have that sanity as well. And often I say, people come to me and say, well, I found this, I've made this observation, I see this behavior in the cell population maybe 2 out of 5 times, and its p-value's kind of weak, but it's significant. And I'm always like, nope. Just even if you go anywhere near that, if another lab tries to reproduce that result, do you think they could reproduce it? It will be incredibly difficult to prove that unless you come with some orthogonal analysis that gives this more support. So, don't go for those weak, barely statistically significant findings. Find something else, another thread to pull on in these data sets.
So Sam, let's say you couldn't do any additional data and you just had to write the manuscript with the data that you have. Then I totally agree, you shouldn't go on these. But isn't it fine for the data to be sort of shaky if you have the inclination to follow it up and design a new experiment and just go on that tangent?
I think that's okay. But I think if you're trying to publish something, make claims based on shaky data, it's just not worth it. I mean, we had that in a manuscript a couple of years ago. So it was where we were doing single cell analysis of the mouse small intestine after most of the proximal small intestine had been resected. And we found involvement of a particular signaling pathway, but yeah, from the single cell data, it was, barely significant. You've got so many cells, it kind of makes it look probably more biologically significant than it is.
Mm.
And when we followed up with orthogonal analysis, it was barely significant. And I said,
Okay.
it's just not worth putting it in. What will we gain from this? Is this going to help another group? Are they going to be able to reproduce it? I also think it's really important to publish negative results as well. If we do publish those results, we'll be clear about it. Well, this doesn't look so significant, but not trying to make more of it than there is really.
No, of course. I mean, you always have to be open about, how much signal there is and how much signal there isn't.
Mm-hmm.
I would like to come back to the issue of creativity. And you were saying that an important part of your creativity is during the writing process. But if you think about somewhere more in the middle of a project where you have data that just doesn't seem to make any sense, right? It doesn't look like it's just an experimental mistake, but the signals that you see are not the ones that you expected, and you have to somehow figure out what is going on. Do you have a process, a specific habit or technique that you use in that type of situation?
That's the best situation, right? When the results are completely the opposite of what you were expecting.
Yeah.
And I always, when my student or postdocs come to me and they're like, "Oh, this has gone horribly wrong. My hypothesis sucks. This doesn't make any sense." And I'm always delighted. I'm like, "Oh, this is incredible." First of all, we go back to the original hypothesis. And that's why it's really nice to have a hypothesis in these situations and ask, well, how might this hypothesis really be misleading us here? What are we missing that could help us interpret this data? And then that always goes back to a literature search, right? What have we missed here? How do we interpret this new data? And then you'd have to go back, revise your hypothesis, do additional experiments if you really are so far off and it's such an unexpected result. And then I think sometimes just going to the community and asking them their opinion. Has anybody else ever seen this? What are their ideas? Every so often I'm known to go to Twitter when we see something strange or we don't know what's happening with a particular situation, and the community is incredibly helpful. I love getting results that are completely unexpected and don't make any sense. As long as it's not technical failure. I think that's the first question. Like, have we technically failed here? Did we get the samples mixed up? But then once you've figured all that out, then you can get creative. And that's where the literature helps.
So, Sam, I'm not sure if you actually answered my question, which was, do you have a certain habit?
Definitely checking the literature, reframing the hypothesis if necessary, but just asking yourself, did I miss anything? Was my idea to begin with completely biased? Did I not take something into account? We work with cells. They're always going to do unexpected things, depending on the context as well. So trying to take a step back and ask, why am I seeing this result? What could it mean? How do I try and interpret that? But then developing the new hypothesis and then developing the new experiments to address that hypothesis. That's usually where I go from there. Yeah, I think to kind of illustrate this point, we did the lineage tracing on the reprogramming. So this fibroblast progenitor reprogramming. And the dogma in the field is that this is a stochastic process. So reprogramming initiation and the early phases of reprogramming are thought to be stochastic, that if you have two sisters that receive the same levels of reprogramming factors, one of those sisters will successfully reprogram, but the other sister will go off and die or turn into a different cell type. And that is just not what we were seeing. That's what we hypothesized, but we found that these sisters followed the same reprogramming trajectories. So the process, the early stages of reprogramming looked a lot more deterministic than we were expecting, which was a surprise. And now this is something that has generated new directions. We now focus a lot more on these earlier stages of reprogramming. It's led us to develop new technologies to address these early stages because we have no way of prospectively isolating these cells. So it's led to a lot of creativity in the lab because The experiments are now way more challenging to try and find out what's happening because only 2% of these cells will successfully reprogram. So it's like looking for a needle in a haystack. But it forces you to think, how do we address this in a different way? But also forces you to think about it technically, especially with the single cell lineage tracing. So thinking, is there a technical issue here? Why these cells would look like they're doing the same things? Are we just missing, those other sisters that died? Obviously, we're never going to capture them. And this then has pushed us into going back toward live imaging because we want to be able to capture these cells. We want to be able to see them, see their neighbors. We now are understanding there's a huge spatial component in these reprogramming strategies. So it's leading us to new technologies, new experiments to be able to address these surprising results that we got. So I don't know if that answers your question.
No, it does. It does.
Sam, it's very clear that technology, both experimental and computational, plays a huge role in your work. I want to ask you about another aspect, and this is the language that you use. For example, you talked about cells that are in a dead end, and there are other analogies that we use in how we describe our results. Do you feel that you have a special language that you use with your lab in describing the results, and this also sort of shapes the creative aspect in generating new ideas?
We do, and we talk about this constantly because every now and again that's definitely led us down our own dead end in a way. When people are using these terms, but they have different definitions of those terms, and that's always amazed me. We collaborate with a lot of different groups, and even when it comes down to the terms lineage and clonal, it's surprising, or trajectory. It's really surprised me how people use these terms and assign different meanings to them. So I think it's really important to be able to define these terms. So this is something we take a step back every so often, and because somebody will present in a lab meeting and they will declare something with very defined, confident language, and the group has the confidence to challenge each other, which is really important, and say, hang on a minute, what you just said makes no sense to me if we're using this definition of trajectory or of lineage. And then we'll realize that, oh, okay, so this is how you're defining this term. No, we need to come back to a solid definition of that. And it really surprises me how often we run into those issues.
So language and precise language seems to play an important role, of course. Do you also find that, especially when you're being creative in the discussions with your people, that you use language that is deliberately vague and, using analogies or anthropomorphisms?
Definitely. And I think that when you don't have a concrete answer or concrete results of a particular question, then you can easily slip into those terms. Or if your technology doesn't have the resolution, then you can start adopting terms that will basically cover yourself for this. So you can always go back to it and say, oh, well, we use this term because we didn't have this resolution. So one of the terms I use for our cell tag lineage tracing is because we do lose a lot of lineage information there. So we can only go through 3 rounds of tagging. So I call these rudimentary lineage trees. And I feel like the word rudimentary there covers all the problems that could be highlighted about this technology. I said, well, I've acknowledged this is rudimentary, so it makes it okay. But also, you have that acknowledgement to the experts in the field. But it's a really important word to use for the non-experts in the field because they need to know that we can do better on this. We can go deeper. It's only going to give us so much information. So I think those terms are really important to communicate to a general audience as well, and to not mislead them. So I think that's the specific example I could come up with there.
And Sam, when you're generating the analysis of the data and you're making visualizations, what is the relative roles of aesthetic aspects of the figure? Is it beautiful? Is it clear? And versus this potential that the image has for generating new ideas. So basically, what is the role of visualization in your work?
I put so much energy into visualization. I love to communicate science. I hate being baffled when I sit in a talk and I can't follow it and it's overly complicated. I say this sometimes when my trainees generate figures or they write something and they try and blind you with the science. There's so many technical terms in there. Like, I can't understand what you're saying, and I work with you every day. Like, this is too complex. Your figures are so detailed. Like, try and break them down a bit for a general audience. Try and simplify them, but don't make them inaccurate. And I think that's such a challenge. I also really like, when I give talks, working on visualizations that are specific for those talks. So not just taking the figures from the paper or the preprint or the upcoming work, but generating new visualization for a specific purpose to explain a concept and communicate that to a broad audience. The papers are getting so long and convoluted now that in the main body of the paper and the main figures, keep them simple. There's just so much noise out there right now in terms of the number of papers that are being published. Like, make sure clearly communicate these concepts. And then in the supplement, that's the area where you can really take a deep dive and explain everything and put all your math in the supplement. I know this is probably a terrible thing to say, but just to help people kind of wade through the literature and your complicated figures, put those in the supplement. They're going to be there for the people who are digging into this and for the experts in the field. I think it's getting really challenging to be able to broadly communicate the results, but also accurately communicate them. So we've kind of got this two-tier system, right? You've got the main text of the paper, and then you've got the supplement. And I'm going down a rabbit hole with this. That's how I'm seeing it right now. Also, some of the papers I've been seeing lately and reviewing, and also the papers I edit. So I'm an associate editor at Development. The papers that I'm enjoying are the ones that do this. They kind of ease you into it, give you kind of all the key highlights. And then when you want to get into, how this actually works or the results that support these main conclusions, then the supplement is there. It does make it challenging. I don't know what your thoughts are on that.
Yeah. Well, I actually want to explore more your thoughts about the broader influence of a paper you mentioned. And this also goes back to what you were saying earlier about how a scientist also has a public relations aspect, which perhaps the most tangible aspect is on Twitter. I'm wondering what you see as sort of your responsibility in the role that you play as how science gets communicated. In other words, what is it that we scientists should strive for the public to know about?
Yeah, that's the public communication of the science. It's something I've been involved in on and off. I love tweeting. I love using Twitter. That's where I find a lot of new publications and preprints and ideas. So there's, on one aspect, you are communicating with your direct community. So the single cell genomics community, a lot of people on Twitter there, a lot of active discussion. But then you've got this broader public communication, and definitely something I got into. So there was this stat madness. It's like March Madness, but for scientists. And the lab got involved in this competition, and we were selected to highlight our research. You get on camera and explain our research in the simplest terms. And it was a 2-minute piece. It took me about an hour. Science communicators at the university were saying, nope, nobody's going to understand what that is.
Oh, no.
Make it simple, make it simple, make it simple. So just going through that process again and again and again was really challenging. So this was to communicate single cell lineage tracing via lentiviral barcoding to the general public, and what the benefit of that was in these reprogramming and regenerative medicine strategies. And so in the end, we were really just focusing on the regenerative medicine because it's much easier to communicate the significance of that. Yeah, really challenging to do that.
No, it's challenging, and we're constantly striving to get the public more on board with the science mission. And you doing this conversation with us also helps. As we sort of bring it to a close, we want to thank you for spending time with us.
Yeah, thank you. Thanks. It's been a lot of fun.
Yeah, it was great. Thank you.